My name is Miguel Soares. I'm a principal investigator at the Instituto Gulbenkian Ciência. I lead the inflammation laboratory. So inflammation is a very promiscuous process. It's kind of embedded in almost all the diseases that we suffer from. They can be infectious or non-infectious. For example, they can be sepsis, malaria, tuberculosis. There's a very strong component of these diseases. And we start realizing that we could modulate inflammatory responses in a way that would protect an infected host from the infection. Basically, the infected host would die or survive. But when we look at the pathogen, there was no effect on the pathogen. And we realized that there was a burgeoning field coming from ecology and immunology that stated that some species protect themselves from infection via a mechanism that we call disease tolerance. They don't target the pathogen directly, but they just assume that they're infected and they adapt to the fact that they're infected. And we realized that the mechanisms that we were working on were exactly those ones. These mechanisms are really controlled at the level of metabolic pathways, by which we use metabolites to produce energy from different sources. And we study these kind of complex mechanisms in a whole organism, and we use mice because mice are mammals and they have the same organs as we have. So they have lungs, they have kidney, they have liver, and we're now understanding that these metabolic responses that provide disease tolerance to infection, actually they rely on inter-organ communication processes, and we're becoming exceedingly interested to understand how does the fat communicate with the liver or with the lungs or with the kidneys in the context of infection. By changing these metabolic processes, we can really make an infected host, in this case a mouse, live or die from an infection without touching or without interfering with the pathogen. So basically what we found was that there's a gene that is expressed in transplanted organs that produces carbon monoxide and protects the organ from being rejected. Me personally, the most rewarding is to discover the unknown, to realize that we have discovered something that could or has importance to humankind, and it was completely unknown. I think that gives you a sense of thrill of understanding the biology of this process that lasts for a very, very long time. I'm not very often outside of the lab, but when I'm outside of the lab, I like to travel. I spend a lot of time in Alentejo, which I really enjoy, getting together with friends. That's about it.